Hey guys, hey biologists out there, particularly my students. This is your first round of biology notes in the flipped classroom. So these notes are going to be done at home or during the task working time that's offered in class. Please make sure that you look at the agenda to see when that class time is offered, and definitely get used to making plans as to when you're going to tackle these notes. They're never very long, they're less than 15 minutes, so they're very, very doable in a short amount of time. Plus, you have the added benefit of watching them again, especially for particularly maybe some difficult content or something like that. And you can pause it so you can finish writing. You can rewind it. I mean, there's just a lot of benefits to having this in video format. Of course, I am available during the class if you have any questions. Now, to start out, you guys, there's a cool Cornell note-taking method that is out there, and you may know about it already, but it's a suggestion that you take notes using this method. You do not have to if you have different ways of taking notes in your own way that you learn best. I mean, ultimately the goal is to learn best, but the Cornell note-taking is tried and true and has worked for many across probably the world. Anyways, on your sheet of paper, you're going to pretend that this is your sheet of paper. Boop, 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 this big little area. So you're going to have notes taking up the bulk of the sheet, the bulk of the page. This is where you're going to record definitions, terms, you know, any of the concepts that you see in the slides. Do not necessarily feel that you have to write everything down. In fact, it's best to abbreviate, you know, and put it in your own words or whatever. However you want to take that down, this is the meat right here, okay? And it's not during class, it's just when you're listening to these notes. Now, the cues over here on the left, these are going to be some of the drawings that you might see on there, a diagram. It might be a quick little memory word for yourself. But most importantly, this is going to be a place where you can write questions, okay? And then your questions, since you're watching this when I'm not necessarily next to you, your questions should be addressed the next day in class for sure from me, and then you can write answers down to those. Also, it's a nice idea to put main ideas here as well. You'll get in a rhythm, especially if you take on this way of note-taking. Down here at the bottom is just a great way to summarize what you've written, and there's a couple ways you can do this. You can do it as soon as you're done with that page, or you can do it a couple of days later, maybe as a quick little reminder. This is what I remember about this page, and write that in there. Quick top main ideas, that's what you're looking for down here, not a lot. So, without further ado, Unit 1, Segment 1. Unit 1 has 3 segments, and so this is the first one. You will be taking a practice quiz and doing other assignments besides the notes, and you will always have a final quiz over each segment. Chunk it so that you can own it that much easier. The main idea for this segment is what is science. We're going to do some word-borne, word-play in class, but let's talk about what is science. What it is, obviously it's a goal to understand the natural world. That's why science is around. We're innate in that we ask questions. I mean, we're constantly asking questions about what we see and hear and do in the world. So it's an organized way of using evidence to learn more about that natural world that we live in. But we base it on evidence, and that is where science is defined. It's really looking at, is it testable? Is there evidence to support it? And if not, then it's not science. Science is a body of knowledge built up after years of using the process. So it builds upon each other. You see that there's no scientist ever anywhere in the world that works by themselves. They have to use the network of scientists in the same area of study that they are to find out what are you seeing? Are you seeing the same things I am? And blah, blah, blah. Now, quick look at what it's not, and we'll talk more about this in class, but this would be the non-natural world. This would be questions of faith. This would be things that are not testable, that don't have evidence to support. that would be what science is not. Traits of a great scientist, you guys, some of you, these might be traits of a good learner also. You're skeptical, you question often, you have an open mind so that you can look at the existing data, the data that you may have and know, but you're open-minded enough to look at new ideas that are coming into play. It may allow you to revise what you know. So that actually makes up a good learner too. But scientists are very open-minded. They look at concrete evidence. They look at the direction that it's pointing as far as what conclusions are made. They have, but they also need to recognize the limits of science. If it's not testable, then we need to tread carefully there. All right, I'm going to start out with kind of like an evolution of how science kind of has come, you know, the evolution of science, I guess. Where did we start? You know, like prior to the mid-1600s, people obviously didn't know the things that we know now today, and they looked at the world around them and they saw living things just show up. And the question of where did life come from arose by many people, and they could not, without understanding the cell and understanding life itself, they would see things just appear. So for example, maggots, which turn into flies, would always appear by meat. So did the meat make the maggots? Mice always showed up around grain. Did the grain spontaneously produce mice? What you're looking at, that little ball over there, that's dung. That is poo from the cows, and there are beetles that will show up there because they actually use that little piece of dung there for their life cycle. They lay an egg inside it, and the egg turns into a larva, and larva eats the dung for nutrition. But they didn't know all that life cycle business, and so they looked at these things and thought, spontaneous generation, these things are coming about, bringing about that life that is around it. In the 1600s, mid-1600s, Francesco Redi actually came up with an experiment to actually test this hypothesis. So Redi's experiment, I think it was 68, he thought, okay, I'm going to take the meat. We think that the meat is being used to derive life in the form of flies, maggots, and then flies. So I'm going to see if that's really the case. So he took flasks, he left one to open air, he sealed one completely, and then he put mesh over the third and left it there. And over time, he noticed that there were flies on the inside of the jar that was open to the air and on the outside. But the one that was sealed, the one in the middle, he didn't notice any flies around at all. And in the mesh, the flies were only on the outside and not on the inside. So there was something to this. If this was the same piece of meat, he controlled all his variables, the same, size of jar, the same chunk of meat, size of meat, everything, all conditions were the same except for he controlled what was at the top. And he noticed, since it's the same meat, it should have the same life. And he did this over and over and over. He did it over a series of years. And he pretty much, with this experiment, said, no, guys, meat is not producing flies. The flies are actually coming to the meat and laying eggs. But for a very long time, they believed, people were very much caught up in the spontaneous generation. Well, Pasteur knew about Redi's experiment and went ahead, and actually some scientists in that time were arguing, well, wait a minute, is air required? Is air required for life? Is it coming from the air? You had it sealed off, the meat was sealed off, so maybe air had something to do with that. So there was a couple other scientists that did a few other tests that you'll read about in some of your other assignments, but here, as far as the test is concerned, Pasteur is who you need to know about. And he went ahead and proved that air was not what was making the living things. So in order for him to prove that, he had to actually not seal one of the flasks. Like Redi had the middle jar sealed completely, whereas there was no air exchange. So here, if he allowed air in, but not necessarily the flies, then he could prove that flies were not just coming from the meat. So instead of meat, he used broth. And broth that will allow, left out, it's kind of like chicken broth or chicken noodle soup or whatever, you're going to end up with some bacteria growth and things like that, microbes. Because at that time, too, they are also noticing that there were cells there in the world. So the broth was boiled so that there was no living things. He made this neck opening that prevented flies from being able to fly in, but yet had air exchange. And then he had another one that was open to the air and did not have a curved neck. And what he found was that the one that was open did produce the microbes, and the one that was open but had the neck curve opening, preventing any microbes from getting in, he noticed there was no microbe development in there, which again goes to the idea that there isn't spontaneous generation, that it is not life coming from non-living things. Life is actually coming from other life. Okay, so anyways, the goal of, we'll work more with those scientists and other scientists as well, but the goal of science is really just to study the natural world in a very organized and specific way. Why? So that we can trust science, so that when a scientist speaks of their findings, we can trust that. Biology specifically is a study that seeks to understand the living world, not the non-living world necessarily, but the living world and aspects of that. And actually that is where we stop. So segment 1, done.